Hello, everyone. Thank you for attending and welcome to the third annual Gene and Cell Engineering Summit presented by GenScript. This summit will feature in-depth discussions and research presentations on the crucial gene synthesis and cell editing technologies that are transforming scientific and biomedical research today. Before we start, we would like to provide some housekeeping items. All talks will be available on demand following the summit in the Labroots auditorium for the next 12 months. Please use the chat box to ask questions during the Q&A session after each talk. We offer a variety of prizes for the winners on the leaderboard. The leaderboard will be locked down at 5:20 p.m. Eastern Daylight Time, and we will announce the winners at 5:25 p.m. Eastern Daylight Time during the awards ceremony. The exhibit hall and virtual booths are open. You may visit during the break between 1:30 to 1:50 p.m. Eastern Daylight Time and 3:45 to 4:05 p.m. Eastern Daylight Time. For event support or help, please visit the help desk. Click Career Opportunities in the help desk for open positions at GenScript to learn more about employment opportunities. Please don't forget to take our feedback survey linked on the kiosk in the lobby and share on social media using hashtag GCE Summit. Shortly after this introduction, we will welcome Dr. Ray Chen, President of the Life Science Group, who will offer some opening remarks and introduce our keynote speaker, Dr. Dimitrios Wagner, Head of R&D at Berlin Center for Advanced Therapies. Here's an overview of the agenda for today. Following the keynote presentation, we will have talks 2 and 3 between 12:15 and 1:30 p.m. Eastern Daylight Time. The first section will then be followed by a 20-minute break. Subsequently, we will have talks 4, 5, and 6 between 1:50 and 3:45 p.m. Eastern Daylight Time, followed by a second 20-minute break. After that, we will have talk 7 from 4:05 to 4:40 p.m. Eastern Daylight Time, followed by our closing keynote presentation presented by Dr. John Zuris, Director of Editing Technologies, Editas Medicine. Kay Chuang, GenScript's Head of Marketing, will conduct the award ceremony, including the announcement of the 2023 Molecular Cloud Distinguished Research Awards, as well as our leaderboard winners. Lastly, Rumen Bagov, GenScript's Head of Segment Marketing, will present the closing remarks to conclude our summit. Please take a few minutes to become familiar with our lobby. There is a welcome video at the top area of the lobby. There is a feedback survey that we would love for you to participate in, linked to the kiosk. Take another moment here to become familiar with our auditorium. This is the room in which all presentations throughout today's event will take place. You can access a specific presentation simply by clicking on its title on the auditorium screen. We would also like you to become familiar with the exhibit hall. There are 5 booths for different services. In each booth, you will find files for supporting resources and ongoing promotions. You may send us a message if you are interested in a particular service. Lastly, we have our networking lounge. In this room, you can connect and network with your peers and colleagues. We want to take the time to thank the GenScript team for making this year's Gene and Cell Engineering Summit possible. And lastly, I would like to introduce career opportunities here at GenScript. For more information, please find the flyer in the help desk. And now I would like to welcome Dr. Ray Chen from GenScript. Dr. Chen, over to you.
Hello, everyone, and welcome to GenScript's annual Gene and Cell Engineering Virtual Summit. We're very excited to have you joining this event for the 3rd year in a row. Yes, for the past 3 years now, and GenScript has convened this summit, which brings together leaders working with cutting-edge approaches to develop novel and life-changing gene and cell therapies. Thank you so much for joining today. And for those who may not have familiar with GenScript, I would like to introduce a little bit. For the past 2 decades, GenScript have lived a shared purpose. It started as an idea for a better and easy way to make genes for researchers. Like all powerful ideas, it evolved into our mission to become the world leading enabling platform in serving sciences by providing reliable, high quality, and innovative reagent services and instruments. This mission has become what I have been passionate about. Now, we have built a diverse family of services and product portfolios across a wide range of existing and emerging life science research and development areas, from early research stages of target discovery to the preclinical and even clinical stages, particularly for gene and cell therapies. We make genes, peptides, proteins, develop antibodies, and make sgRNA and non-viral payloads, and mRNA, and more. Now, we are more committed than ever to continue developing and building our platform capabilities and capacities around the globe to better serve innovations and development in gene and cell engineering globally. We're excited that we have expanded our 350,000 square foot GMP facility for the sgRNA and non-viral payloads for CRISPR genome editing and cell engineering. We're now reliably delivering mRNA from Seattle, and we further expanded our molecular biology and protein facilities in Singapore and Piscataway, New Jersey, US. We do hope that we can be more and more helpful. And thank you again for joining this summit today. We have a full agenda of stimulating presentations. We will be hearing from academia and biotech leaders who will share their work, and starting from our keynote speaker, Dr. Wagner, and for the new strategies for immune cell reprogramming. And we will also be hearing about the new viral and non-viral generating tools and platforms, target discovery in Parkinson's disease, metabolic pathway engineering in microorganisms, engineered cells as model system for drug discovery. They're all exciting topics, and there must be very good presentations that will be shared. And also we'll have several fun activities planned out throughout the booth, and we will have support throughout the day as well. GenScript team are always available to assist you for any questions you may have. So thank you again for joining, and I will hand the stage to our moderator, Dr. Claire Zhou, and for introduction for our keynote speaker. Claire?
Hi. Great, thank you very much for the introduction. Hi, this is Claire Zhou. I'm the product manager for CRISPR services here at GenScript. Thank you all again for joining our third GCE Summit. And it's my great honor today to introduce our opening keynote speaker, Dr. Dimitrios Laurin Wagner here. Dr. Wagner is the head of research and development unit at the Berlin Center for Advanced Therapies, BeCAT, or Charité, the Berlin University Medicine. Dr. Wagner's expertise is in immunology and gene engineering with a focus on reprogramming of T and NK lymphocytes for therapeutic interventions in cancer, transplantation, and autoimmunity. In particular, CRISPR-Cas9 gene editing and chimeric antigen receptors, as well as related technologies. In today's talk, he will share with us his recent findings on CD3ζ as a novel integration site for reprogramming immune cells with chimeric antigen receptors. Please do submit any questions you may have during the presentation in the question box, and we will answer as many questions as we have time for at the end of the presentation. And with that, Dr. Wagner, please feel free to start.
Thank you very much for the wonderful introduction. It's a real honor to present one of the stories from our lab today as a start for this really exciting program later today. So I'll start off with my potential conflict of interest. So my lab is focused on how we can best use gene editing to create very potent therapies for immune-related diseases. And of course, CAR T-cell therapy is one of the prime examples how genetically engineered cells can be used to treat disease. And my camera is turned off because my internet connection might not be amazing, but I hope you can still hear me good. So CAR T, the concept behind CAR T cell is amazing, if you ask me, because the idea here is that you take T cells from the patient's own body. These are immune cells that are highly capable of eliminating cancer cells, and we teach them in the lab to better recognize cancers, grow many of them, and then reinfuse them where the cells can use the chimeric antigen receptor, short CAR molecule, to find and destroy cancer in the patient's body. As one example from a case report, you can see here a PET scan. So this is a patient on the left side that has end-stage lymphoma. You can see the black dots. This is basically all cancer spread out through the entire body, and only 30 days after CAR T-cell treatments, most of the black dots have disappeared from the body. The residual black signals are really just normal signals that we would expect in such a PET scan, and this patient has been cured of his disease. Now, CAR T-cell therapy is amazing, and we have multiple commercial products available, but on the other side, while these therapies are effective, they're also very expensive. And since most CAR T-cells that are available commercially are personalized products, which means that we actually have to source the T-cells from the patient themselves and then make the product, send it back, there are also time delays associated with this treatment. Now, out of an academic development perspective, in particular GMP-grade retroviruses, which are currently the gold standard for gene transfer to create these CAR T-cells, are a major roadblock because they pose significant costs and time delays at an early development phase. However, also, if you consider that you might want to overcome the problem of autologous and personalized manufacturing, if you would like to move towards off-the-shelf T-cell products, then retroviral gene transfer is not enough. You will need additional genetic engineering. So we set out to find non-viral solutions to create CAR T-cells. Looking at what potential opportunities for gene transfer would be, and we exclude retrovirus because we don't want to use this, and we also exclude transient approaches like mRNA or transient episomal vectors, then there are basically two main technologies that you could use. On the one side, the transposase technology, which basically is using transposase enzymes to semi-randomly integrate DNA transposons into the genome, or target-specific gene editing. Transposase technology has been around for longer, but unfortunately also made some bad news. A couple of years ago, we unfortunate to hear the news that 2 out of 10 patients in a clinical trial that were treated with CAR T cells generated with an hyperactive version of piggyback transposase actually developed lymphoma out of the CAR T cells. So this really tells us that semi-random integration and in particular strong exogenous promoters at the wrong sites can still pose the risk for oncogenic transformation, and therefore we decided to move on to use gene editing as a platform to develop non-viral CAR T cells. So at the center for gene editing for gene transfer is still the programmable nuclease, and our favorite programmable nuclease is the CRISPR-Cas9 system from Streptococcus pyogenes. The Cas9 system was engineered to be a two-component system, which constitutes of, on the one side, a Cas9 nuclease protein, and on the other side, a guide RNA, where we can basically exchange a small target sequence called spacer sequence to make this complex specific for almost any part in the human genome. Now, once this complex is introduced into cells, it can find the specific location it was designed to be specific for, and then this complex induces a double-strand break, and this is where gene editing starts. Normally, this break would be repaired by this cell through non-homologous end joining, which is a DNA repair mechanism that is error-prone. And if we guide a CRISPR complex to a protein-coding gene, we can really use this to enforce small insertion and deletions and use it to knock out. However, to create a CAR T cells, we will have to exploit another DNA repair mechanism that is called homology-directed repair, or short HDR. Here we co-deliver donor templates that these are DNA constructs designed to look very similar to the DNA surrounding the break site. And these DNA donor templates can be used by the cell's endogenous repair mechanism to repair the break through a proofreading function. And by adding high amounts of donor templates, we can basically trick the cell to insert new artificial sequences at the desired locations. And this is really what we set out to do in the lab. The DNA template can be delivered in different forms. The current gold standards to create CAR T cells is actually not a non-viral solution. It is a virus called AAV. You can use AAV to basically deliver these donor templates to T cells. However, AAV faces the same challenges as retrovirus since they are complex to manufacture and hard to procure in a cost-effective way. Then there are also synthetic solutions like double-stranded or single-stranded DNA, which can come in different flavors like circular or linear. And we set out to use and optimize a non-viral gene editing platform on DNA because it has very good stability and can be manufactured either enzymatically or in E. coli very efficiently by many different providers. So luckily, when we started our lab in 2020, there were already nice publications that describe that actually this is possible. Namely, this landmark study by the lab of Alex Marson, which showed that if you co-deliver the CRISPR-Cas9 system as a ribonucleoprotein, so basically the recombinant Cas9 and a synthetic guide RNA as a complex, together with linear double-stranded DNA donor formats, you can induce very efficient homology-directed repair in T cells when you deliver all these components to activated T cells. The group also showed that you can use this to transfer transgenic TCRs, and we really wanted to adapt and optimize this method to create CAR T cells. So Jonas, the first, an MD and my first PhD student in the lab, set out to do this. And first he designed a DNA repair template, which allows us to specifically insert a second generation CAR sequence into the first exon of the TCR receptor alpha chain constant gene. So as a consequence of this desired gene edit, we would have cells that instead of making TCR alpha chain, make a truncated TCR alpha chain protein and a CAR. And through this genetic engineering, we are able to basically redefine the specificity of the T cells towards CD19, which is an antigen expressed on most B cell malignancies, and eliminate the T cell receptor because the truncated TCR alpha protein is not stable enough to form a full T cell receptor complex. So how does the workflow look? Usually we source T cells from peripheral blood out of the mononuclear cell fraction. We activate the T cells in the presence of cytokines and polyclonal stimuli for 2 days. And at that time, the cells start to proliferate extensively, and we transfect them with RNP and DNA. And then 2 days later, we analyze them by flow cytometry. And example pictures are shown here. If you have mock electroporated cells that didn't receive the RNP or the DNA template, the T cells all express the TCR CD3 complex on the surface. Here you can see it as FAX plots on the y-axis, but they're unable to express the CAR. If we deliver the RNP, we can induce a knockout. If we co-deliver RNP and the double-stranded DNA, we now have this new population that pops up in the lower right panel, which is basically T cells that express a CAR but no TCR. We went on to optimize the different parameters in this procedure, and we found that, of course, DNA has a major impact on the gene editing outcome. If we increase the amount of donor templates at the time of transfection, we increase the amount of template that could be used for HDR, and we can see higher knock-in rates. However, this comes at a drastic cost of cell survival because the DNA is toxic at high amounts. So if you ever want to, start working in this field, it's really important to really find the optimal dose for your transgene and location where you want to insert, for example, a CAR using non-viral gene editing. In his first paper, Jonas showed that we can, on average, knock in a second-generation CD19-specific CAR in the TRAC locus of T cells at approximately 20 to 25%. However, you can also explore small molecules to really favor homology-directed repair. Here, we collaborated with IDT and used their ALT-R HDR enhancer version 2, which allows you to really boost knock-in rates drastically. We also explored a drug cocktail that could be used to prepare T cells for the electric shock and being flooded with all the DNA. And unfortunately, this drug cocktail was not able to reduce the toxicity of the procedure as intended. However, we found that it nicely synergizes with the ALT-HDR enhancer to further increase knock-in rates. The next big question is, of course, now we are able to make these cells, but how well do they perform in a model where we let them fight against cancer? In this case, we used a leukemia model in immunodeficient mice. So here, NRG mice that are immunodeficient are grafted with a leukemia cell that also expresses luciferase. 4 days later, we inject the same number of CAR T cells, and then we monitor the leukemia outgrowth by bioluminescence imaging. And this was done in collaboration with the lab of Renate Strippeke back in Hanover back in the day. Then what you can see here is the results from this experiment. In the first column, you can see bioluminescence images of mice receiving only the tumor, and you can see that over the 5 weeks, we see a gradual increase of the leukemia signal, and in the end, the mice actually have to be terminated because of this leukemia outgrowth. Both CAR T cells that were generated by the TRAC knock-in, so non-viral gene editing, and the conventional lentivirus transduced CAR T cells are able to slow the leukemia progression in this model. And at least in 2 out of 4 mice in this experiment, we saw that TRAC replaced non-virally gene edited CAR T cells might even be a little bit better than the lentivirus CAR T cells. So as a short summary for this first part, I hope I can convince you that non-viral gene editing with CRISPR-Cas9 and synthetic double-stranded DNA template is an effective means to reprogram T cells, and also that knock-in rates might be improved by HDR enhancers. We've also tested transient DNA sensor inhibition as a mean to prevent toxicity. However, we found that it only had limited impact on the T cell survival, and it could improve a little bit the knock-in rates. And I think this is an interesting avenue of research to identify more potent drug cocktails that prevent toxicity by non-viral vectors. Last point, I've shown you that non-viral TRAC-replaced CAR T cells display comparable or maybe even better anti-leukemia performance than lentivirus transduced CAR T cell products. Now, we had this wonderful tool, and we wanted to ask, is TRAC really the best integration locus for CAR transgene? And to answer this question, we turned on the regular CAR structure. For everyone who's not familiar with this, a CAR usually consists of an outside portion, a binder that interacts with the tumor-associated antigen. This one is directly fused in a single molecule with a hinge domain that provides some distance from the cellular membrane, a transmembrane domain, co-stimulatory domains, and in the end, most conventional CARs have the same effector domain, which is the CD3ζ domain, the intracellular part of the CD3ζ gene. So, of course, when we do TRAC knock-in, what happens is that we will insert the entire genetic information for this CAR with all the different components and introduce a transcriptional stop site. And this allows us to create CAR T cells as shown before. So we reasoned that theoretically the CD3ζ gene is also a gene that is expressed in T cells. So maybe we could just use this as an alternative locus to drive our CAR expression. And as you can see in the next plot, we are able to basically integrate full CARs into that locus and get an expression. However, the more interesting thing for us was really to aim to integrate only the missing sequence to in vivo assemble a full CAR. To this, we transferred a truncated CAR transgene. So basically just the missing components, which is the binder, the hinge, transmembrane, and co-stim, and fuse it directly in line with the endogenous CD3ζ gene, thereby alleviating the need of approximately 1/3 of the transgene insert needed to transfer. We can see by facts here that actually the CAR is efficiently expressed. However, the important question is, does this truncated CAR transgene also convey efficient cytotoxicity in these now newly reprogrammed cells? And this is an important question because of this. If we integrate the same truncated CAR sequence into the TRAC locus, we are able to also express a CAR. However, these CAR T cells are unable to confer any cytotoxicity. That is in the TRAC locus. Now, if we use the endogenous CD3ζ locus, we can nicely rescue this loss of cytotoxicity by basically exploiting the endogenous CD3ζ gene. So now truncated CARs inserted in this particular locus, the CD3ζ gene, drive T cell cytotoxicity in a CAR-dependent manner. We next wanted to look at, how do these CAR T cells look in comparison to TRAC replaced or lentiviral transduced CAR T cells. And one thing that directly pops into your eyes is on the left side, you can see that the CAR expression on average is a little bit lower than in the TRAC locus and also lower than in the lentivirally transduced CAR T cells. When we challenge these CAR T cells with tumor cells, we see that the regulation of the CAR expression is very similar to the TRAC locus. However, we start from a lower baseline level, and actually we also lose a little bit more after the stimulation. However, after 2 to 3 days, it comes back up to baseline. In contrast, lentiviral transduced cells are unable to downregulate the CAR expression, but rather upregulate the CAR after initial antigen encounter, and that might be a reason why they perform worse in preclinical models. So due to the lower CAR expression, we found that they're a little bit worse in short-term cytotoxicity assays, and we wanted to rescue that. So we introduced a small modification to the transgene to really rescue this expression. It's now called CD3 TRUNC-CAR opt. So this is really a small sequence that we insert, and this now increases the CAR expression to a level that's similar to TRAC or maybe even a little bit higher. Then next, we performed CAR T cell stimulation, where we put the CAR T cells together with tumor cells. Then we use brafaldin A to block the Golgi apparatus, and then basically look for all the cytokines that are produced by the T cells. And we look for TNF-alpha, IL-2, and interferon gamma, key anti-inflammatory or key genes that are important for anti-tumor effect. And you can see in the next plot, if you focus on the middle row, where T cells were exposed to NALM6, so CD19 positive cells, the black and the dark gray parts mean that the cells produce 3 or 2 of the cytokines, and these are cells that have high anti-tumor function. And you can see that in the TRAC-CAR, we have a lot of it. In the CD3ζ TRUNC-CAR that was not optimized, it's a little bit less. However, after the optimization of the sequence and the higher CAR expression, we also have a rescue in this CAR-mediated cytokine production. And this is now shown for CD4s, but we've also seen exactly the same trend in CD8 T cells. Now, the interesting question is not, okay, there's a small difference in the short-term functionality, but we wanted to know how do they do long-term. So we compared the different CAR T cells in serial tumor re-challenges. And what Jonas does for that is he takes CAR T cells and mixes them 1-to-1 with leukemia cells, then lets them fight against each other for approximately a week, reanalyzes the cells, takes the remaining CAR T cells, readjusts the co-culture from one CAR T cell to one tumor cell, and then repeats this procedure week after week. And what you can see if you compare the products is that first, all of the 3 non-viral gene-edited CAR T cells expand massively over these serial challenges. And you can see if you look at the amount of CAR T cells, we start between 20 and 40% CAR-positive cells after a single stimulation. Basically, all the T cells left in the culture are CAR-positive. And this is really not lost over the different stimulations. Of course, an important point is, can we learn anything about their fitness? And to this end, we analyzed expression of inhibitory receptors that are commonly associated with exhaustion. And we were able to find, and that's actually quite intriguing, and we don't have a good explanation for it yet, that the CD3ζ locus, regardless on the lower or the higher expression, seem to make the cells a little bit better. And cells after 4 re-challenges actually expressed a little bit less inhibitory receptors than TRACA T cells. Now, this is all in vitro. Of course, we next investigated in the same in vivo model that I presented before. Again, NALM6 leukemia cells, again, 500,000 CAR T cells injected 4 days later, and then we look for the leukemia outgrowth. And here you can see something very fascinating. All of these cells are able to prevent the leukemia outgrowth. However, we can nicely see that the optimized CD3ζ trunk CAR condition is actually the one that performs best in this assay. And this experiment is still running to date, and still 4 out of the 5 mice are alive, while in all other groups, the mice have disease due to the disease. So as a small summary for the T cell part, this basically means we have a new way to make very potent CAR T cells that basically only requires transfer of truncated CARs, which might pose also additional safety advantages. Next advantage is that the CD3 CAR knock-in approach is also compatible with other cell types. Wage in the lab tested this in Tregs and gamma delta T cells, and Clemens in the lab looked at whether we can use this to also redirect NK cells. And NK cells are particularly interesting if you consider off-the-shelf approaches. And NK cells are also a cell type that doesn't express TRAC, so the TRAC gene at all. So TRAC doesn't work in these cell types. And now we have a new non-viral gene editing approach that might work in those cell types. So let's look how these cells do. First, NK cells are an immune cell type that is more part of the innate immune system. It can execute various different functions, and we know that it's not only important to protect us from transformed cancer cells, it can also directly interact with virally infected cells, and they have a very interesting system how they recognize and spare healthy cells. And that is the reason why NK cells from foreign donors do not lead to alloreactivity. Many groups, and I'm showing two excerpts here from work by the Rizvani lab from MD Anderson, have shown that you can use CAR molecules also to enhance the anti-tumor function of CAR NK cells. And I really like you to point to this wonderful landmark study that showed that you can use CAR transduced natural killer cells to effectively fight CD19 positive lymphoid tumors, either autologous, but also they've shown for the first time that you can also use allogenic CAR NK cells that work effectively and don't cause any toxicities, at least in the few patients that they've treated. So now if we want to use CD3ζ as an expression locus, we kind of have to look, okay, it is expressed. It also has a role and function in NK cells. And we know that CD3ζ is an important activating signal transducer for stress receptors, such as NKp30 or NKp46. And these receptors have been implicated in the ability of NK cells to target and recognize cancer cells. And the other important point is that the CD3ζ was long believed to be an essential part of the CD16 complex. And CD16 is the receptor on the surface of NK cells that makes them able to perform antibody-mediated cytotoxicity. That means NK cells are able to recognize cells that have antibody bound in them and then induce cytotoxic action. So potentially, This is an important function of NK cells because this could mean we can give them a CAR, and we can also use monoclonal antibodies to target different antigens on the tumor and avoid, for example, antigen escape variants. So the first question we had, in the T cells, we observed that if we edit the zeta locus, the TCR complex is gone. And the next question is, well, what happens to the CD16 complex, where the zeta chain and another gene called FcR gamma are the main contributors? So we first wanted to do a knock-in or a knock-out of CD3 zeta and figure out whether the CD16 expression and thereby the ability of NK cells to perform antibody-dependent cytotoxicity is impaired. And here's some flow data from Clemens, and you can see in the top row, first of all, after knock-in of a GFP in the endogenous CD3 zeta locus, most of the cells actually lose CD3 expression. In the second row, you can see GFP expression, and we have knock-in rates with this non-viral technique of approximately 14–15%. And then the lower row is the important point here. CD16 expression between mock electroporated and these non-viral gene-edited NK cells is basically different. There's no downregulation of CD16. It rather seems that CD16 might be a little bit higher on the cells that are manipulated. So what does that mean functionally? So we assess the ability of these different zeta-edited NK cells to perform ADCC. Here we took JACO1, a CD20 positive cell line, and incubated that cell line with different antibodies, either rituximab, anti-CD20 antibody, or a control antibody. And in theory, once the antibody binds to the JACO1 cells, it should help the NK cells to specifically kill them. And this is what you can see in the first summary graph in the middle. At increasing amounts of rituximab, we see an increase of the killing of the JACO1 cell. When we use a control antibody, we really only see the background activity that the NK cells have towards this partially NK-resistant cell line. So this means, zeta knockout and the GFP knock-in both didn't change anything about our ability, about the NK cells' ability to execute this function. We also looked at other canonical functions of NK cells, and that is namely how well they can kill different tumor cells. So here on the left side, you can see that the different edited NK cells are able to kill an AML cell in K562s in a dose-dependent manner, and also NALM6 on a base level. However, a very important thing is that either by knockout or knock-in into the zeta locus, we don't change their reactivity towards allogenic healthy cells, which means that we don't believe that this approach makes these NK cells alloreactive, so they hopefully are as safe as normal NK cells would be. So next, we wanted to study whether CD3 zeta-edited CAR NK cells are also able to exhibit enhanced cytotoxicity towards CD19 positive cancer cells. And that is a tricky thing to do because the NK cells in itself, they already have a very high background activity towards most cancer cell lines. So what we did here is we tried to figure out whether there's any preferential killing. So we used the NALM6 cell line that is CD19 positive, and we generated a CD19 knockout mutant. So we have two cell populations, one cell that expresses our antigen of choice and the other one that doesn't express it. Then we mark the two antigen positive and negative cells with different fluorescent dyes and basically do a co-culture with 3 cells. And then we look whether one of the 2 cell types is targeted in a preferential manner. With primary NK cells, where we had around 5% CAR knock-in, we can see that there is, at increasing amounts of NK cells, a dose-dependent advantage towards the killing of the CD19 positive cells. However, both cell types were very efficiently targeted. Since the knock-in rates are not that good with the CAR NK cells, we can't really judge this, what this means, because there's only very few CAR positive cells in this assay, and that we can see something is already good. But to see whether in an enriched format this also works, we use the NK92 cell line. So this is an immortal NK cell line where we were able to do a CAR knock-in and then also enrich for these cells. So in this assay, the CAR NK cells had a purity of 90 or more %. And we compared 3 different NK cell lines, mock electroporated NK cells, CAR NK cells generated by insertion in a safe harbor locus AAVS1, and the Zeta trunk CAR condition. And you can nicely see that the CAR actually significantly enhances the CD19 CAR-dependent cytotoxicity in this model. So we already come to an end from what I wanted to show you here. And I think the 2 main points that I wanted to make is, on the one side, that a non-viral gene editing with double-stranded DNA template is inefficient. And I can tell you it's also a cost-effective gene transfer method in primary human T cells that could be used to generate different CAR T cell products. And that the CD3 gene editing strategy is maybe a new approach to not only redirect T cells, but also NK cells with CARs, so that these cells could be used to enhance anti-tumor efficacy, either in autologous or even allogeneic approaches. As a little bit of outlook, I've already shown you that in certain cell types like NK cells, the knock-in rates were not as good as in the T cells. So we recently formed a new EU consortium called GeneTiger that will try to compare the impact of the different donor formats on the efficacy of gene editing, but also on how well the cells do. And here we also include AAV as a donor template modality. So we really want to figure out, if we use gene editing to create CAR T cells, what is the best way to do this? Are there any fitness differences when generated through any of those different technologies? And in particular, single-stranded DNA, I would like to highlight this, might be very interesting in cell types that are very sensitive to double-stranded DNA. In our hands, double-stranded DNA was very toxic in NK cells, and it would be very interesting to test whether single-stranded DNA might allow us to increase knock-in rates in NK cells to make the CD3 zeta CAR NK approach even more amenable to NK therapies that rely on peripheral blood or cord blood as a cell source. As one additional point, of course, I hope that I could convince you that the knock-in into the zeta locus or the TRAC locus are nice ways to create a CAR T cells that has reduced risk for TCR-mediated graft versus host disease. However, that is only one step to create a very good CAR T cell product for an allogenic off-the-shelf use, and to make an allogenic CAR T cell product better, to help it persist in a patient that also has an immune system, we will have to think about other means to improve it. And 2 obvious strategies would be to eliminate or modulate the expression of MHC class 1 and 2, which are the primary reasons why foreign cells, allogenic cells, are rejected in immune-competent hosts. What you could do is, for example, you can use CRISPR to knock out the beta-2 microglobulin gene, or you can knock out the C2TA gene, which is the master transcription factor necessary for MHC class 2 expression. And what the consequence of that is that you will need to install multiple edits in a single cell. And if you want to use this clinically, it has to be as easy as possible, so you don't want multiple manipulations. And if you do all of these manipulations, knock in and these 2 knockouts at the same time, this could induce a lot of different translocations. And if you are afraid of that, and like us, want to prevent that, please check out the paper that we recently published from Victor Glaser in the lab, where we showed that you can actually combine DNA break-free gene editing technology called base editing with non-viral knock-ins to create multiplex edited CAR T cells that have a minimal amount of translocations and hopefully are safer for clinical use. With this, I would like to thank you all who are in the meeting today, or who look at this talk later on demand, for your time and attention and listening to us. I would like to thank my wonderful team in the lab that made this possible. I tried to highlight their names. I think it's important to highlight Jonas, who was really the first and also one of the main drivers of many of our non-viral gene editing programs, and Clemens, who did all the work on the NK cells. I would also like all my collaborators and supporters, mentors in Charité and BIH, and also our external collaborators who help us address new challenges. And of course, this wouldn't be possible with funding, so thanks to our funding agencies. And with this, I'd be happy to take any questions that you have in the chat or live.
Hi, Dr. Wagner. Thank you very much for the great talk. I think you definitely shared a lot of great events in the field, especially the non-viral engineering of the immune cells. So we'll now start a live Q&A sessions. And if you haven't submitted your questions yet, or busy, listening or trying to understand the talk, now is a good time to start putting in your questions in the question box. And also, I guess with a couple questions already coming in, let's start a Q&A section. So I think the first question is, do you see any similar benefit of the DNA sensor inhibitor you showed in your data on different donor types, such as plasmid DNA or single-stranded DNA?
That is a great question. If you look in the paper that we published last year, you can see what kind of DNA sensor inhibitors we tried. And we actually took a mix to be able, that might be useful not only for linear double-strand templates that we used it for, but also for single-stranded or plasmid. However, we never really tested it with these different donors. So we haven't used it with plasmids. We also haven't used it with single-stranded templates. But we did try to use the same approach in NK cells, and there we saw a similar picture. It was not as good as we hoped. So we hoped that it would be like a black and white situation where we do the pretreatment and then just all the cells survive. It was really not that good. In NK cells, the survival benefit was a little bit better than in T cells, so it might be something interesting to explore for sensitive cell types. However, I believe so far we haven't really cracked the right cocktail, or we just don't know all the DNA senses that cells have to prevent the toxicity of the templates fully.
Great, and I think a question very close to that. So one of the attendees is asking, can you explain why is double-strand DNA toxic to the cells?
So in addition to the fact that there are maybe more sensors like CGAS, AIM2, these are all sensors that might be expressed in the cell types and can bind double-stranded DNA, it might also be contributed to a DNA damage response. So maybe the loose ends or the blank ends of the linear DNA are recognized by the cell as DNA breaks, and it frantically tries to repair it, and that induces stress to the cell. The other point is, from a physical standpoint, double-stranded DNA is just much, much bigger than, for example, a single-stranded template. And if you consider what we do, electroporation is really just blasting holes in these cells and then flooding them with a lot of macromolecules, right? RNP, DNA that might also form bigger complexes together. So there might also be just a physical attribute of double-stranded DNA that might be better with maybe plasmids or other supercoiled formats, where maybe the molecules that you transfer are just smaller and thereby less toxic.
Sure, and I think with Dr. Wagner and GenScript, we're also working on testing different formats of payloads, and as you mentioned, the loose end of the double-strand DNA might be one reason to cause the toxicity. So with our, close end, the Gen 1 double-strand DNA, you also have obtained some data to potentially show that it would lead to like toxicity, or any data you can disclose to the audience here?
So in general, we've used different gene-scrapped materials, and what we found that the double-stranded DNA that we received from GenScript was surprisingly effective at low amounts. And hopefully we can sum up that data in a paper or a preprint soon.
Great. So I will move on to a little bit more questions regarding the editing efficiency. So one question is, you showed about 20% editing efficiency. Is that something clinically significant?
That's a great question. In principle, it is. If you think of the poster child for CAR T-cell therapy, Emma Whitehead, right? So that received a lentiviral transduced CAR T-cell product. The CAR T-cell products that are used commercially, they don't have 90% transduction rates. Many of them actually have much, much less. And if I'm well informed, then Emma Whitehead's product had only 10% CAR-positive cells in the cell product. In the clinic, you see this a lot with commercial products, that they have varying transduction rate, and it all seems to work quite well. Of course, all the CAR T-cell products are always adjusted for the amount of CAR-positive cells that are given. So if you have a low CAR expression percentage, that means that you will have to transfer overall more cells. So of course, it is interesting for anyone who works on this to make it as efficient as possible, to just optimize production.
Great. So more questions coming in. We got quite a few questions now. So this one is, do you have a rough timeline on when you will be bringing this to the clinic?
That is great. I mean, my timeline is always much, much shorter than the reality. So if it would be after me, then maybe late 2024, we would have the first non-viral gene-edited CAR T-cell product close to clinical trial. However, of course, in an academic center, this is always, we need to get funding to do this, and we also need to organize our capacities well. So let's see, maybe end of next year, maybe a little bit later.
Got it. And then I think the next question is, I've read that the AAV8 is more efficient in transferring therapeutic genes. I want to know why you choose AAV6 instead of 8 or other variants.
That's a great question. So the different AAV serotypes are specialized on going into different cells. I'm not an AAV person, so I don't know by heart. I don't know if AAV8 would be better for T cells, but at least in the field, the most commonly used AAV for knock-ins in T cells. Now I'm saying T cells is AAV6. Most groups, the group of Matthew Portois in Stanford, the group of Michel Sadeline, basically everyone who does AAV-based knock-ins uses recombinant AAV6. So maybe serotype 8 is very efficient in another cell type, or maybe you are up to something very interesting.
Great. I hope that answered the audience question. And then a couple more CD3ζ related question. So first one is, does introducing CAR transgene into the CD3ζ gene impact any neighboring genes? Was there any RNA-seq or other assays performed to test this?
Yeah, it's a great question. We haven't done this experiment. And I don't know what the result would be, because of course the CAR transgene itself also has a biological effect on the cells. So it is a good idea to do this. It would be really good to have the right controls for this kind of experiment, to kind of subtract any effect that would be dependent on the CAR. But otherwise, this is a great experiment that we should do and hopefully will do in the future.
Great. And then another one is, have you compared the CD3ζ trunk CAR knock-in efficiency with the track locus knock-in?
Yes, we regularly do this in the lab. In general, the TRAC locus seems to be a locus that is very, very good for knock-ins, and the CD3ζ locus seems to be a little bit worse. However, by reducing the size of the transgene, we are able to, on the one side, we have to insert a smaller piece. That's always easier. And then due to the smaller size of the transgene, we can also use more DNA. So that's a thing with linear double-stranded DNA templates. The bigger your template is, the more toxic it is, and if the smaller you make it, the less, the more template you can use without killing your cells. So for us, that particular locus, due to locus-specific effects, I'm sure, seems not to be as good for larger transgenes. However, for the small ones that we use, it's as good as TRUC.
Well, so have you tested anything larger than like a CD19, as you shown here, or I guess what's the larger size you've made it possible into the CD3ζ side?
So the CD3ζ, we never tried more than the full CAR, which is basically including the poly-A transcriptional stop signal. It's around 2 kb insert. And the 2 kb insert usually goes in at a rate of like 15 to 20%. So it's lower than TRUC, where we usually get the same sequence in approximately 20 to 30, or even in good experiments, maybe even more than 30. Yeah.
Yeah, and also I think because the NK cells, especially you show the data that how it could be targeting NK without really having a TRAC option there is
Yes.
a great benefits.
For this, we need good single-stranded DNA quality and very high concentrations. I believe that might be a solution for the NK cells. At least that's my hope.
Yeah, I think we'll definitely take this offline, discuss collaboration opportunities on this, as well as we do provide pretty long and high quality single-strand DNA as well.
Wonderful.
I think a couple more questions. Maybe we'll take two more. One is, have you considered other different Cas9 proteins instead of different Cas proteins instead of Cas9?
Yes, definitely. You in the publication from Victor Glaser, which I mentioned, we actually optimized gene editing with Cas12a for the TRUC locus. We've also optimized the Cas12a enzyme for the zeta locus. So in particular, Cas12a from Acetoaminococcus species are an interesting modality because they have a different cutting pattern, so that it cuts distant to the palm. It was always proposed that this might increase knock-in rates, and in our hands, it really seems that Cas12a might be better. However, the breakthrough really came through the new Cas12a mutants, Cas12a ultra, that was published by IDT together with editors, and you will definitely see more data on how Cas12a can be used for knock-ins by John Zuris later, as who will do the last keynote presentation of this day.
Yes. So one last question we'll take is, I guess, how can we leverage the low immune system in cancer and high immune system in autoimmune disorders to develop a novel approach for combating these disorders?
Yeah, that is it is a great question. So I think the answer is already in our body, right? In situations where we have a low immunity towards cancer, we can use T cells that we engineer to be hyperactive against a given cancer with a CAR. So we can use conventional T cells, give them a CAR, or we can use NK cells, give them a CAR. So the cells can go into the tumor, lyse a big portion of it, and also reprogram the immunosuppressive microenvironment. That's for cancer, and for disturbed immune system in autoimmune disease. I believe there's another cell type called regulatory T cells, so an immunosuppressive T cell type that might be optimal to also balance and rebalance autoimmune diseases. So here, of course, it's interesting with the regulatory T cells. What do you direct it against? Like, can you make a regulatory T cell specific for the tissue where the autoimmune disease is active? For example, multiple sclerosis, an autoimmune disease of the brain, right? Could we make a regulatory T cell that can go into the brain and prevent the destruction there? And this is definitely an active interest of our lab. And I think there's a lot of exciting things ahead for using the same principles that we use for CAR T-cell therapy in cancer, also for chronic autoimmune diseases, which are as terrible to treat from a physician's standpoint as severe cancer.
Great. And lastly, I just want to, I guess, close with a question, or like to have your insights or comments on, as I know you spend quite some effort in, I would say advocating, but testing non-viral approaches for allogeneic CAR T development or NK cells or else. Where do you see the current bottlenecks lies in, further pushing more non-viral products into the market or into the clinic?
I personally think that we are already beyond the brink. We've already seen wonderful studies from China and from the US where people have used non-viral gene editing based system to make very potent products. There's a non-viral CAR T-cell product that was originally made in China that exploits the PD-1 locus and a CD19 CAR. They have seen very good responses with this. Then in the US, they have used the non-viral system to make a neoantigen-specific T-cell receptor therapy, and they've also seen that this is safe and can be manufactured. I think now it's just a matter of time. I'm very confident that we will see more non-viral gene-edited cell products very, very soon, because many people agree on the advantages. And let's hope that there will be, more resources, cheaper reagents. That's of course a very important thing for the academic side, and then we can test more products.
Great, and I definitely think your hopeful wishes, we will wish those come true, as well as we do spend quite some I guess investments in providing more non-viral reagents for supporting this CAR T or other therapeutic development on GenScript side. So yeah, I think that's all the questions we'll take due to time limit. Thank you, Dr. Wagner, and for all the questions we haven't got time to answer regarding either getting a certificate or question about getting an internship at Dr. Wagner's lab, we'll try our best to connect you after the talk today. And thank you again for joining. I don't know if Dr. Wagner, do you have any final comments or anything else you want to share with the audience here today?
No, I thank you very much again for inviting me to speak here. Thank you everyone for listening. I hope this makes you excited about trying some of these techniques in your own lab, and maybe you'll be in suspense for the paper to come out. So would be great. If you have any more questions, reach out. We're always happy to help.
Thank you, Dr. Wagner, and thank you all again, the audience, for joining the talk as well. We do have our exhibitor booth open as well, so please feel free to browse through and chat with our online representatives there. And thanks again. Hope you enjoy the talks coming up soon. Thank you.
Thank you. Bye-bye.
Thank you.